Mitochondria, often called the powerhouses of the cell, are far more than simple energy generators. These dynamic organelles play central roles in regulating cellular metabolism, redox balance, immune signaling, and programmed cell death, processes that become increasingly important with age. As such, the decline of mitochondrial function is now recognized as a hallmark of aging and a contributor to age-related diseases ranging from neurodegeneration to cardiovascular dysfunction. Yet in long-lived individuals, LLIs, including centenarians, mitochondria appear to defy this trend. These individuals often maintain remarkably efficient mitochondrial activity well into late life, with lower levels of oxidative stress and sustained energy output that support cellular resilience. This mitochondrial preservation is increasingly seen not as an accident of aging, but as a key enabler of exceptional longevity. In typical aging, mitochondria accumulate damage to their membranes, proteins, and DNA, especially mitochondrial DNA and tDNA, which lacks many of the protective and repair mechanisms available to nuclear DNA. This damage contributes to increased production of reactive oxygen species, ROS, a byproduct of mitochondrial respiration that can damage cellular components if not properly neutralized. The feedback loop between mitochondrial dysfunction and oxidative stress accelerates tissue degeneration and contributes to systemic inflammation, insulin resistance, and cognitive decline. However, studies of centenarians and other LLIs reveal a distinct mitochondrial profile. Mitochondrial function in these individuals is characterized by greater efficiency in oxidative phosphorylation, the process by which mitochondria generate ADP, the body's primary energy currency. This efficiency means that centenarians produce sufficient cellular energy while generating fewer harmful byproducts like ROS. In many cases, LLIs exhibit lower systemic markers of oxidative damage, such as lipid peroxides and oxidized proteins, compared to age-matched individuals with typical aging trajectories. This suggests that their mitochondria not only function better, but are also better maintained and protected throughout life. One factor contributing to this advantage may be the capacity for mitochondrial quality control, specifically mitophagy, the selective recycling of damaged mitochondria. In LLIs, this process appears to remain more active and effective, allowing cells to remove dysfunctional mitochondria before they trigger inflammation or apoptosis. At the same time, mitochondrial biogenesis, the formation of new mitochondria, is maintained at a level sufficient to replenish and sustain energy-demanding tissues such as the brain, heart, and skeletal muscle. This turnover and renewal capacity is critical for cellular health and may underlie the preserved physical and cognitive function seen in many centenarians. Genetic factors also shape mitochondrial aging trajectories, particularly variations in mitochondrial haplogroups, clusters of mtDNA variants that are inherited maternally and differ between populations. Certain haplogroups have been repeatedly associated with increased longevity in epidemiological studies. For example, haplogroup J in European populations and haplogroup D4A in Japanese individuals have been linked to greater mitochondrial efficiency, reduced ROS production, and enhanced resistance to apoptosis. These haplogroups may promote more stable mitochondrial membranes, improved coupling of respiration, or greater resilience to metabolic stress. It is notable that these haplogroups do not act in isolation. Their influence on aging is shaped by interactions with nuclear genes, lifestyle factors, and environmental exposures. The same haplogroup may confer longevity benefits in one context and neutral or even detrimental effects in another. This highlights the importance of gene-environment interactions in shaping mitochondrial aging and the need to consider individual and population-level variation when designing interventions. Lifestyle factors play a substantial role in modulating mitochondrial health across the life course. Physical activity is one of the most powerful known stimulators of mitochondrial biogenesis, particularly in skeletal muscle and brain tissue. Regular endurance and resistance exercise improves mitochondrial function by upregulating the expression of genes involved in energy metabolism and antioxidant defense, such as PGC-1A. Similarly, dietary patterns that reduce mitochondrial burden, such as calorie moderation, intermittent fasting, or diets rich in plant polyphenol, appear to promote mitochondrial efficiency and stress resilience. These practices may work, in part, by activating AMPK and sirtuin signaling pathways, which enhance mitochondrial maintenance and limit inflammation. Interestingly, the offspring of centenarians often show mitochondrial traits intermediate between their parents and the general population, suggesting that both inherited and environmental factors contribute to mitochondrial health. This transgenerational effect implies that mitochondrial optimization may begin early in life and accumulate over time, influencing not just lifespan but long-term health span. The role of mitochondria in aging is no longer viewed as a passive decline in cellular energy. Rather, it is an active and modifiable process that appears central to the success of LLIs. By preserving mitochondrial function, whether through genetics, behavior, or emerging therapies, these individuals maintain the capacity for cellular adaptation, regeneration, and defense, all of which are crucial for surviving into extreme old age without major disability. As interest grows in interventions that promote healthy aging, mitochondria have become a focal point for therapeutic strategies. Compounds that support mitochondrial biogenesis, reduce oxidative stress, or enhance mitophagy are currently under investigation for their potential to delay or reverse aspects of biological aging. The long-lived provide a natural model for what these interventions aim to achieve: sustained energy, reduced cellular damage, and prolonged physiological function. In many ways, to understand how to live well beyond 100, we must first understand how to keep our mitochondria young.